I will talk about the BCMA-targeted therapy with CAR-T. The problems with the CAR-T is that it's a new technology. It is not readily available, and it has its own toxicity, meaning that it might cause severe cytokine release syndrome or might cause neurotoxicity. And therefore, AL patients who are very fragile and very delicate in terms of both treatment responses and their heart condition, this may not be the right treatment for them. So we started by preclinical laboratory data where we wanted to check if CAR-T was feasible for AL cells. And what we did is first we measured the amount of BCMA expressed on plasma cells derived from AL amyloidosis patients, and we were able to show that it is expressed. It's less, its expression is less pronounced than in multiple myeloma, but it's certainly expressed, and obviously enough to be sensitive to the CAR-T cells. And we made autologous CAR-T cells from patients in co-cultures with the plasma cells. They were able to eliminate the plasma cells, and therefore we were thinking to move on to a clinical study. In this respect, we've designed a clinical study for myeloma patients, and we added our amyloid patients to the study. So in general, we had a phase 1 study using our CAR-T. This is an in-house CAR-T, meaning that it was designed in the laboratory of Professor Polina Stepansky at Hadassah Medical Center in collaboration with the Bar-Ilan University, and it's a construct that was designed over there and utilized by the laboratory to produce our own academic CAR-T. And this is a phase 1 study where we took mostly myeloma patients, some amyloid patients, and it was a 3x3 phase 1 study where we were starting with very low doses of cells, 150 million cells per patient. And after seeing safety, going over to 450, and after safety analysis, going over to 800 million cells per patient. And in terms of the general safety in all population, it seems safe, so we're continuing into this phase 1b with another 40 patients planned to be checked in this phase 1b. And during the phase 1a, where we had 20 patients, in general, we did not see too many side effects, but I will state them in a moment. Within the study, we had 5 amyloid patients in different cohorts. One of the patients was also a compassionate patient. He had MDS, and we couldn't enroll him to the trial, but he received the CAR-T after receiving the authorization of our Helsinki committee. So these 5 patients were treated with the CAR-T. 1 patient at the dose of 150 million cells, 2 patients at the dose of 450, and 2 patients treated at the dose of 800 million cells. The results were I'll state them in a moment, but in terms of safety, we did have some CRSs, the cytokine release syndrome, and this was shown mostly with congestive heart failure. So we cannot really know if these amyloid patients did have CRS or was it just congestive heart failure exacerbation. In terms of other side effects, they were pretty much manageable. And in terms of response rate, I must say that 4 of these patients had cardiac involvement, and some of them severe cardiac involvement in terms of high proBNP of 7,000 or 4,000 and so. And also there was one patient with just renal amyloid, and he had severe proteinuria for years. So after receiving the CAR-T cells, about a month after, almost all patients were in complete remission and MRD negativity. And in terms of organ response, it was striking to see that almost all patients had organ responses, and these happened very fast. So in terms of cardiac proBNP, it went down by sometimes more than half within 3 months. And in terms of the proteinuria of the renal amyloid patient, it actually disappeared, something that he never had ever in his life. So this was very striking in terms of the fast response and the organ responses seen. As for durability, that's a big question. We don't know yet. We don't have enough time to say if it will be durable for a long time. Most patients were maintaining their remission for at least 9 or more months, but the trial has been going on for 1 year roughly, so we still don't know how long and how durable the responses will be. Certainly at the 800 million dose, it will be a relatively long durability.